there is not enough time for antigen-presenting cells to take it up. So there will not be a good immune response. So chemical stability is also important. Foreignness, it should be foreign to the animal. Whatever it is you are injecting as an antigen. Dose, don't think if you inject a bigger dose, you can produce a lot more antibodies. So that's not right. Again, small dose may not induce enough antibodies. Why we are saying very big dose may not induce good level of antibody, that might cause tolerance, and your immune system will not work. If you inject too much, immune system might be tolerized and it may not work. Complexity, again, we said chemical stability is needed for the antigen-presenting cells to take it up, but if it is too complex, we put some materials into the body, right? But they are not degraded in the body, are not taken up by the antigen-presenting cells, like such as silicone or implants, whatever. They are not taken up by the immune system, so they cannot produce any antibodies. In some cases, that's good, because when we put some implants into the body, we don't want immune response. Okay. And size of the molecules. We do make some DNA vaccine. Just use DNA from the virus and use that as a vaccine. It may not induce good immune response, but we now know we can put it with a carrier and make it more immunogenic. So size is also important, and that's route of administration. So that's about talking about antigens. But again, within the antigens, there are thymus-dependent and thymus-independent antigens. When I said thymus-dependent, most of the antigens will induce antibody formation by B lymphocytes, but those B lymphocytes require that help from the T helper cells. T helper cells producing those cytokines will make the B lymphocytes to produce antibodies. So again, without those cytokines, without that help from T cells, B cells will not produce good level of antibodies for most of the antigens, which we now call as T-dependent or thymus-dependent antigens. There are thymus-independent antigens. Few of them with long repeated chains can induce antibody formation from B lymphocytes without that help from T helper cells. There are few. These are bigger repeating antigens, the repeating antigenic molecules, such as lipopolysaccharides, for example, can induce antibody formation from B lymphocytes without much help from the T helper lymphocytes. So did I make that understandable? T-dependent and T-independent antigens. For most antigens, we need that help from T helper cells. Okay. Looks a little bit complicated, but what saying the same thing. B cells need that help from T cells to produce antibodies. That's all it's saying. The picture is showing here, the B cell is recognizing this virus, but it depends on this help from the T cell through the cytokines that they produce. So now it differentiates into a plasma cell and then secretes those immunoglobulin molecules or antibodies. Again, same thing. They need that help from the T helper cells to produce those antibodies. Okay. What exactly these antibodies do? We are talking about antibodies can neutralize viruses or bacteria, but directly they do opsonize. Don't forget the term opsonization. They coat that organism or the microbial organism. They coat, the antibodies coat. That's opsonization. That helps in phagocytosis. And they also activate complement. Which pathway is the one involved in antibody-mediated activation of complement? That's the classical pathway. Remember those three pathways? We talked about classical pathway, alternate pathway, and lectin pathway, right? The classical pathway is the one that depends on those antibodies. Antibodies bind to that microbial organism and then activate the complement proteins in the blood. Okay. Slide showing the same thing again. I don't have to repeat this, except you are seeing the B cell is differentiating into that plasma cell with a lot of high endoplasmic reticulum, which is the source for the antibody formation. Okay, so that's the antibody forming cell actually. Okay. All that I want you to get out of this slide again is the help from the T cells is needed. T helper cell help is needed for these B cells to produce the antibodies. Okay. Don't worry too much about other aspects of that slide. Now coming to, we talked about immunoglobulins or antibodies. Sometimes we abbreviate them. Immunoglobulin is IG. Okay. There are 5 types: IgG, M, A, E, and D. And these alphabets are given based on the type of the heavy chain they have. They have heavy chains and light chains, but depending on the type of heavy chain, you will see in the next slides. It could be gamma, mu, alpha, epsilon, or delta. Based on the types of the heavy chains, they are named as IgG, M, A, E, or D. So 5 immunoglobulin molecules. IgG is a basic immunoglobulin molecule. And we will see again details about this, but so are IgD and IgE. They look a little bit similar, except IgE has a little bit more structure here. You will see that later. But IgA is a dimer. It has 2 basic immunoglobulin molecules, which you are seeing here and here, and it's joined together here. But IgM is a pentamer, meaning it has 5 basic molecules joined together by the J chain here. So we'll come back again to these 5 immunoglobulins molecules. That's the same picture again. IgG is a basic immunoglobulin molecule. So is IgD and IgE, whereas IgA is a dimer. It has 2 basic immunoglobulin molecules joined together by a J chain. And IgM is a pentamer, meaning 5 basic molecules joined together by a J chain. That's pretty much the same. I don't want to spend any time on that slide. Coming here, again, you see that IgM is a pentamer. We said it has 5 basic molecules, which has, you should move this to here actually. It has 5 basic molecules, and the heavy chain type is mu. That's why we call it IgM. And because it has 5 basic molecules joined together by J chain, its molecular weight is much higher as compared to other molecules. Whereas IgA, we said it's a dimer. Again, you see that molecular weight is higher than IgG or IgG and IgD. And again, heavy chain types, IgG is gamma, IgA is alpha, epsilon, and delta. Okay, basic molecules, IgM has 5, IgG has 1, IgA has 2, and IgD has just 1. Okay. What is the functions of these antibodies? We said they can neutralize. But first one is toxin neutralization. You all took tetanus shot. What do you expect? You took a tetanus shot, you made antibodies, right? Made antibodies against the tetanus toxin. Tetanus is caused by what bacteria? Clostridium tetanus. And whenever there is a wound, bacteria multiply at that site, but they produce the toxins. That's causing the problem. It's not the bacteria directly, right? So you have to neutralize the toxin. So the tetanus shot you took will make you produce the antibodies, and they will neutralize, if at all you get the infection, have tetanus, the toxins will be neutralized by these antibodies. Okay, microbial binding. They bind, antibodies bind to the bacteria, virus, and prevents the attachment of that virus to the specific cell receptor. Because the antibody is able to bind to that specific cell receptor, it will not allow the virus to bind and enter the cell. Virus must enter the cell and then replicate, and then produce new virus particles. But antibody will prevent its attachment to that receptor and prevent the virus to enter the cell. But antibodies can directly neutralize the virus's bacteria. Cytotoxicity against virus-infected cells. We said cytotoxic T cells kill virus-infected cells, but antibodies also help in that cytotoxicity. We talked about opsonization, we talked about complement fixation or complement activation by especially classical pathway, don't forget that. And antibodies, but it's mainly IgE antibody can trigger the mast cell to degranulate and release its content, which is mainly histone. So IgE antibodies can trigger mast cells to degranulate. And coming to the basic structure of the immunoglobulin molecule, like IgG, it's a basic immunoglobulin molecule, right? It has one heavy chain on this side and another heavy chain. There are 2 heavy chains and there are 2 light chains, okay? And this heavy chain is usually about 50,000 Daltons molecular weight. So 2 heavy chains together will be 100,000 Daltons. And the light chain, each one is about 25,000 Daltons. So the whole molecule is about 150K Daltons. But it has few other molecules here, like disulfide bonds that are binding the heavy chain with the light chain. And there is also what's known as the hinge region. Hinge region here will add a little bit more to the molecular weight, apart from the heavy chains and light chains. So that's the basic immunoglobulin molecule. Again, remember, 2 heavy chains, 2 light chains, And the antigens like virus will bind at this site. So we call this antigen binding site and antigen binding site on either side. So IgG molecule has two antigen binding sites. Now if I ask the question, how many antigen binding sites are there on an IgA molecule? If you're not dozing off, we should be able to tell quickly which immunoglobulin molecule.
2? We would have 4.
4. IgA is a dimer.
Yeah.
It has two basic molecules, so 2 on each side, so that will be 4. And now we should be able to tell IgM has how many binding sites? 10. That's right, because it's the pentamer, 5 basic molecules, so it should have 10 binding sites. Okay, antigen binding sites, the amino acid composition is highly variable from molecule to molecule. Animals produce millions of types of antibodies. Question?
Can you just point out the heavy and light chains?
Okay, we will come back, but a basic immunoglobulin molecule like IgG has heavy chain. So there are 2 heavy chains, okay? And it has 2 light chains, okay, on either side. We will see another slide next one, okay? And as I was saying here, at this antigen binding site, the amino acid composition is highly variable from molecule to molecule. Animals produce millions of types of antibodies depending on which antigens are inducing its formation. And they will bind only to the antigen that induced its formation. So they should be highly variable. So that's why they are very highly variable here at the antigen binding sites. Importance of that will come back. But the light chain here is bound to the heavy chain with a disulfide bond, okay, which are shown here in the red colors. And between the 2 heavy chains, there is what's known as the hinge region. The hinge region allows the 2 arms of the antibody to freely rotate and bind to any antigens. So that allows that antibody molecules to bind quickly any antigens coming into the picture. Again, this slide shows again the same thing. Heavy chain here, 2 heavy chains and 2 light chains. And there are 2 kinds of light chains. They call it lambda or kappa. Which is given in your lecture notes, but it could be either lambda type or it could be kappa type light chains. Heavy chain could be gamma, mu, alpha, epsilon, or delta, based on that we classified in our other one. But here there are a lot of disulfide bonds, you can see. And these regions, the antigen binding sites or regions, are highly variable within the amino acid composition. And within that variable region, there are hyper-variable regions, again, that confers a lot of diversity. Again, you should remember you are exposed to millions of types of antigens, and you will produce millions of types of antibodies, and they are different from each other. And there is a hinge region here that will allow the 2 arms of the molecules to freely rotate, and that's located, and this is important. Within the heavy chain, look at this closely. There is a variable region of the heavy chain here, and all this is the constant region. The amino acid composition in this part is very constant. It's only variable in this region. Okay? And within that constant region, there are three domains, or three segments, which we call as constant region of the heavy chain. So that's why it's called CH1. There is CH2, constant region of the heavy chain 2, constant region of the heavy chain 3. So there are three segments there. Between CH1 and CH2, we see that hinge region. Hinge region is located where? It's between CH1 and CH2. Okay, again, remember for questions in the box, where does this antibody bind to complement binding region? That's CH2 region. It binds to the complement proteins that are present in animals' blood, and those bind at the CH2 region. Whereas it binds to what's known as FC receptor, such as on neutrophil, for example, at this region, which we call now as CH3. Okay, any questions on that? Remember again, where does it bind to complement? We should be able to say CH2 region. Where does it bind to FC receptors on the neutrophils? It should be a CH3 region. Hinge region is located between CH1 and CH2. Any questions on that antibody molecule? So that's a basic immunoglobulin molecule such as IgG. Okay. Again, that's the same thing we have seen.